All right, so we are going to try something different here with the slides. I made a slideshow, and we'll work with this in this lecture. And this chapter, chapter 12, that we're going to start talking about now, is related to modern applications of genetics and microbes. So we're going to take a look at some genetic engineering type things and how it is that we can use the information that we've already learned in these chapters to apply to this type of thing. So first we're going to define a couple of terms. So biotechnology and then genetic engineering. So biotechnology is, as it says here, the science of utilizing living systems to benefit humankind. So if we break that word apart, right, so bio is living thing, and then technology. So we're using technology or newer things, utilizing science to benefit biology, to benefit living things and living systems. So and then we have genetic engineering, which is altering an organism's genome to achieve desirable traits. So oftentimes we're using genetic engineering in biotechnology. So that's how we're able to benefit humankind utilizing these living systems, is by doing genetic engineering, by changing an organism's genome. This image that's down here is a PCR machine, and that's one of the things we're going to be talking about in detail, is how to go about doing polymerase chain reaction, as that's a common technique in genetic engineering, which is encompassed in biotechnology. So first, we're going to start by talking about what is recombinant DNA technology. So recombinant DNA, this is where we're going to take a DNA sequence and then we're going to manipulate it. So this is, again, related to genetic engineering. So we're taking the DNA sequence and we're going to alter it in some way or change it in some way. So this is actually, this whole process of doing this is called recombinant DNA technology. And then what is called recombinant DNA is the DNA that contains new combinations of genetic material. So if we think about, perhaps you've heard of genetically modified organisms, for example. Genetically modified organisms are an example of organisms that have recombinant DNA. So where it's been modified genetically, right? So we've gone in, we've modified the DNA, we've placed that DNA back in an organism, and now that's a genetically modified organism, and therefore it has recombinant DNA molecules because it contains new combinations of genetic material. So it's introduced into a host organism. Then that host organism is actually called transgenic. So the host organism that has DNA from different species. We kind of commonly or colloquially, when it's related to food, call it genetically modified organisms. And that's also similar in term here to transgenic. So a good reason for us to do this, so if we think about it, oftentimes I think when genetic engineering comes up or genetically modified organisms come up, a lot of times people think of a lot of the negative stuff that's out there about genetically modified organisms. But prior to it kind of being on the scene as far as intake for food, really we've utilized it in the way of bacteria that can produce insulin using a plasmid. So this is so that we don't have to go around killing a whole bunch of animals and taking their insulin, which is how it used to be. There's porcine insulin, which means insulin that comes from pigs. And so then they used to slaughter pigs and then, take out all of their insulin, and then that would be used for diabetics, typically for the type 1 diabetic, who is unable to make insulin or make enough insulin to live. Now we can put the DNA for insulin into a plasmid, and we can put that plasmid into bacteria, and then that bacteria is going to make many, many copies of that insulin for us, and then we can, clarify that, we can centrifuge it, just make sure that we have only the insulin, get rid of the rest of the cells. So we lyse the cells, we get the insulin, purify it, and then we're able to use that instead. And that's what this image is down here. So we're talking about, they're showing that there's a human cell here. Then we have the DNA inside the nucleus. We take out the insulin gene. Then we take that insulin gene and we put it into a plasmid. So here's the plasmid that would go into a typical bacterial cell. We cut it right here. And then we insert that plasmid. Now this is called recombinant DNA. That recombinant DNA is then put into a bacterium. Now that bacterium is called transgenic because it contains the recombinant DNA. And then that bacteria is going to go through binary fission and make lots and lots and lots of copies of itself. And every single time it makes a copy of itself, as we know, it copies all of the DNA as well as plasmids. So then we have our recombinant bacteria have the ability to secrete human insulin. So then we can collect the insulin, we can continue growing these organisms, making sure that we keep the culture healthy by adding different nutrients, by taking away wastes, and then we can continue them producing insulin. So this is one way that we've used this, and this also gives us these couple of terms, so recombinant DNA and transgenic. So how do we go about doing this? The recombinant DNA part, making the recombinant DNA. How we go about doing that is through what's called molecular cloning. So molecular cloning is this set of methods that's used to construct the recombinant DNA and then facilitate their replication in host organisms. And so we're going to break this down further into further steps here, but this is the general overview process of this. So here first we utilize our restriction enzymes, and again we're going to go over this in detail, but we utilize what are called restriction enzymes, and restriction enzymes are enzymes that go in and they cut DNA at very specific sequences. So we can go in and we can use the restriction enzyme to cut both the foreign DNA and the plasmid vectors, and that's actually what we see in the image on the right here. We have our foreign DNA, which in this example, we're saying the foreign DNA would be like our human DNA that has the insulin gene in it. So we would cut that DNA. We would also cut the plasmid vector here. Once both are cut with the same restriction enzyme, they cut at the same spot in both the foreign DNA and the plasmid because it goes by sequence. The enzyme will search through the DNA and it will cut it every time it sees its certain sequence. So we cut both of those. Then we have ligation, and this is where we're going to paste the foreign DNA chunks that we have cut using those restriction enzymes and paste them into the plasmid, and they're going to fit. You can see they mentioned sticky ends here, which we'll talk about in more detail. They fit because they're cut with the same enzymes. So you can actually look at the C's and the G's and the T's and A's that they have here, and you can see that they match up perfectly. So then we are introducing that recombinant DNA into the host organism. So basically we've manipulated this DNA outside of the organism, so in vitro, and then we're going to take that and we put it back into the organism. So we put it into the bacterium. This is, again, oftentimes bacteria. So we put it into the bacterium, and then we're going to talk about this in detail, but we can do this blue-white colony screening in order to make sure that we get bacterial colonies that we can then mass produce. We can culture more of. So let's take a closer look at each of these steps. The first step was utilizing restriction enzymes. So restriction enzymes are also called endonucleases. And these endonucleases are naturally occurring enzymes. We actually get them from viruses and bacteria. So we get them from viruses because they utilize these enzymes in order to chop up host DNA so that the host DNA doesn't work anymore. So remember when a virus goes into a cell, remember it's going to chop up the host DNA so that the host DNA can't use its own machinery anymore, so that the virus can take over the machinery. When the virus takes over the machinery, then it can use the machinery as much as it wants, and the host is no longer trying to use the machinery. We can see the same thing from bacteria, but in a different way. So we've already spoken about bacteria that have particular enzymes that they can utilize, like casinase, or different proteinases, lipases, phospholipases, those types of enzymes, but they're specific enzymes to DNA. And so, again, this is so bacteria can go in and we can chop up the DNA, and then it's not going to be a problem. The reason that bacteria also have enzymes that cut up DNA is because when we get to the end of the semester and we're talking about the ways that our immune system tries to fight off bacteria, we actually have these things called nets, N-E-Ts, and our body, one way that it tries to kind of catch bacteria and prevent it from further developing in the body and causing infection is by creating DNA nets, and it really is like a netting of DNA. And so bacteria have developed ways of getting around those bacteria or those DNA nets, and that's by utilizing these endonucleases. So we have gotten these naturally occurring enzymes from the virus, from the bacteria species, and then we produce them, mass produce them, make lots and lots of copies of them, purify the enzyme, and we utilize them in our molecular cloning process. So we can use these restriction enzymes or endonucleases to cut DNA. And it can be then we can cut DNA that we're wanting to put into something, and we can cut the DNA that we're putting it into so that they have the same cuts. And that's what we're going to talk about here. So each restriction enzyme has a particular recognition site. So this is a specific DNA sequence that the enzyme is going to cut. As it says here, it's usually a palindrome. So it's a 4 to 6 base pair length, and it's the same on both ends. So if you look at the image here on the right, you can see that we have our DNA, 3' to 5', 5' to 3', and then if we just look at sticky end cutting to start off with, a restriction enzyme or an endonuclease can come in and it will recognize this entire section. Once it recognizes this entire section, it's going to make the cut on that sugar phosphate backbone. When it makes the cut on the sugar phosphate backbone, you can see here it's showing where it cuts. It cuts in the same spot on both strands. So if you look at this strand here, we have CCTAGG, which is 3' to 5', and it's going to cut between, if we ignore the right side, it's going to cut between the 2 G's at the end of this 6 nucleotide sequence. If we look at the other side, 3' to 5', we also have CCTAGG. It's the same thing, it's a palindrome. And so then again, it's still wherever it finds this sequence of CCTAGG, it's going to cut between the 2 G's on the sugar phosphate backbone. So then what happens is we end up cutting here, and then we end up cutting here, and then that breaks these hydrogen bonds here. And so then we have essentially just cut and broken apart the DNA, and you can see that here. And this particular type of cut is called sticky ends. And that's because we have these nucleotides here that want to hydrogen bond, and they're just basically hanging out there nice and sticky, meaning they are going to try to hydrogen bond to anything that they can find. So sometimes they just go right back and hydrogen bond with their opposite side, and the DNA doesn't end up getting cut. Sometimes then it's just hanging out and it's just looking for something to stick to, which is important because then when we cut our other piece of DNA and try to stick them together, we have sticky ends on both that are very helpful and make it much easier to ligate them. Sometimes DNA doesn't work out that well for us, and so sometimes we have to do blunt end cutting. On blunt end cutting, you can see in this case the endonuclease is going to recognize CCGG, so it's going to cut it between those C's and the G's there in the middle, and on the other side we have our CCGG, and it cuts it there, but then what we're left with is just a straight cut here, and we don't have overhang ends, and so this is why it's called blunt end cutting. There isn't anything that's trying to anneal right now. There isn't anything that's trying to ligate or stick back together. And so it actually makes it a lot more difficult to go through the genetic engineering or the molecular cloning process because those sticky ends aren't out there trying to attach to something else. It's still done, and it's done because the particular DNA that we're trying to cut may not have any sort of palindrome sequence that an endonuclease or a restriction enzyme can find. And so then we're stuck with having to use a blunt end endonuclease. So then what we're going to do is we are going to anneal. So annealing is where we are going to form those hydrogen bonds between those complementary bases. So remember we had those sticky ends, and those nucleotides are out there just trying to hydrogen bond with something else. And so the actual process of forming that bond between that nucleotide that's trying to hydrogen bond and another, nucleotide that's trying to bond is called annealing. So when the two nucleotides with their hydrogen bonds bond together, that's annealing. Now, hybridization is just the general term for the joining together of these complementary single strands of DNA. So when we talk about the process, we're talking about hybridization is when we are taking DNA and we're putting it together. Annealing are the specifics of creating that hydrogen bond between the complementary bases specifically. Now, ligation is when DNA ligase is going to rejoin those sugar phosphate backbones through covalent bonding. So again, annealing is the hydrogen bonds between the bases. Ligation is the backbones through covalent bonding, right? Because those are cut. That's what those endonucleases or restriction enzymes are actually cutting. They're actually cutting the backbones, the sugar phosphate backbone, and then it breaks apart. So those hydrogen bonds break apart. They don't have the strength of those covalent bonds of the backbone because they've been cut. So then when we put them back together, when we hybridize, which is trying to put strands of DNA together, the joining together of the strands, we have both annealing happening, which is those hydrogen bonds forming at the bases, and ligation happening, which is connecting those backbones, so that covalent bond in the backbones. And again, as mentioned in here, the blunt ends are less efficient, right, because we don't have that sticky end. So when we do blunt end hybridization, then it's not as efficient. We don't get as much of a product because it's more difficult, versus sticky end hybridization, which is much easier, and we get a lot more product. So in the picture down here at the bottom, the image, again, this is kind of blowing up the one that we already looked at, but here you can kind of see what they're saying with taking the foreign DNA and cutting the plasmid. And here what they're saying is the plasmid with ampicillin resistance gene. So we're saying, okay, let's take this plasmid that has ampicillin resistance on it, and we're going to add some chunk of DNA from our foreign DNA, and we're going to put it into the plasmid that also has ampicillin resistance, and we'll see why in just a moment. So in the plasmid, the restriction site is in this area here, right? So we're actually going to talk about that in more detail. But we have this area that the restriction enzyme is going to cut, and that's the restriction site. So we're going to cut that, and then we're going to do some annealing and ligating, and then that is part of our hybridization. So the reason I kind of went forward here is because I want to talk about the restriction enzyme sites specifically separately. So what we are doing is we are using plasmids as vectors, right? So something that's going to carry DNA from one organism to another organism, just like we've spoken about vectors in the past, except we're doing this specifically in a laboratory, and that's why it's molecular cloning. So this right here that you're seeing down here on the right, this is called PUC19. And this is a plasmid that has been specifically designed to move DNA from one organism to another organism. And up here, we're talking about scientific supply companies. So these specific companies can build these plasmids and put them in there with specific areas for enzymes to cut. So that's what these specialized properties are. Part of them is that they have specific areas to cut, and they also confer different types of things like antibiotic resistance, for example, or it can be like a fluorescence protein, or it can encode for a fluorescence protein. Things like that that allow you to see whether or not that specific plasmid has then gotten into the organism that you're trying to put it into. So in the example here, it's antibiotic resistance. And in the picture before, remember we had the AMP gene at the bottom, and that's because it had ampicillin resistance. Then you know that if you are trying to put this plasmid, let's pretend this one has ampicillin resistance. Oh, it does right up here. So if you're trying to put this PUC19 into a bacterium, then what you'll do is you'll go through the steps of doing that, which we're going to talk about, but then you can plate it on ampicillin. If you plate it on ampicillin, a plate, an agar plate that has ampicillin in it, the actual antibiotic, then only those organisms that are resistant to that ampicillin would grow on that plate. And so if the bacteria did get this plasmid into it, then it would be able to withstand that ampicillin, and therefore we know if it grows on there that it has the plasmid that you want in it. And then, of course, you can take that and you can make copies from that. So that is one way to know if you got your plasmid inside the bacteria. So another piece to that, which I was starting to talk about, is called the polylinker site, or the multiple cloning site, the MCS. So that you can see here, the polylinker site, or the MCS. The MCS, as it says, is the sequence containing multiple unique restriction enzyme recognition sites. What that means is this has been specifically designed to allow different types of restriction enzymes to cut it in this MCS region, or the polylinker site. You can see here all of these are different types of enzymes, so restriction enzymes, like EcoR1 is a restriction enzyme, XBAL is a restriction enzyme, Hindi3 is a restriction enzyme. So each of these are different types of restriction enzymes that can be used in molecular cloning. So if a person is going to use EcoR1, for example, to cut the foreign DNA, let's pretend it's the insulin gene, they're going to use EcoR1 to cut the insulin gene out of the human DNA. Then they can use this plasmid here and cut it also with EcoR1. If they cut it with EcoR1 here and cut the insulin gene out with EcoR1, then we can stick them back together, hybridize them. And then this particular plasmid is going to contain the insulin gene as well. Utilizing that restriction enzyme, we can stick them back together. Now again, if we do that and then we put it back into the organism, we need to know which organisms got the right thing. We can do that utilizing what's called a reporter gene. So a reporter gene is one that contains the MCS and a sequence that encodes a protein for visualization of DNA insertion. So this is so that you can actually visually see, like I was mentioning, the fluorescent protein. So one example is if it encodes for a fluorescence, then the organism that has that particular plasmid that has the genes to code for the fluorescence protein would then glow. It would be fluorescent under UV light. In this example here, it's saying containing LAC-Z. So if we were saying that this particular plasmid, let's say that this plasmid doesn't have this here because it has it up here anyway, but let's say this particular plasmid has LAC-Z, then around here, right, we would have LAC-Z, the enzyme or the genes or DNA that encodes for LAC-Z. So if this plasmid, before we try to put any DNA into it, any foreign DNA into it, if we were to plate it out on a plate, it's going to be able to make LAC-Z, right? It's going to make LAC-Z because LAC-Z is in it and we haven't done anything to change this plasmid at this point. So if we were to plate it as is, it contains LAC-Z, and if it contains LAC-Z, then it can degrade lactose and XGAL. So if we take this plasmid as is, put it into a bacteria, plate that bacteria on an agar plate that contains XGAL, it will degrade XGAL because it has the LAC-Z gene, and when it does that, it actually turns the colony blue. So the actual bacteria on the plate look blue. Now, if we then want to put DNA, foreign DNA, let's say the insulin gene, into this PUC19 plasmid, then what we're going to do, let's say we use EcoR1 and we cut our insulin gene out of the DNA, the human DNA, and say we cut this plasmid here and we insert the insulin gene into the plasmid. Now we've got a huge chunk of DNA within the LAC-Z genes that are going to code for LAC-Z. When that happens, then the bacteria that has this plasmid in it is not going to be able to make the protein for LAC-Z anymore. And it can't make the protein for LAC-Z anymore because it's been disrupted, right? We have stuck the insulin gene right in the middle of this LAC-Z, and so now it can't make LAC-Z anymore. So in this way, if we were to take that then and plate it out on a plate that has XGAL, it does not have LAC-Z, so it will not degrade the XGAL. They'll grow, but they're going to be white colonies. So they're just going to grow like regular bacteria. They're not going to degrade the XGAL. So then we know that that LAC-Z gene has been disrupted. We know if it is a white colony that we have successfully cut right in the middle of the LAC-Z gene, and we've successfully added our DNA because LAC-Z is no longer present to break down the XGAL and turn blue. So those are some important pieces of our plasmid that we would be using for molecular cloning. So we cut our plasmid, we put in, say, our insulin gene, and then now we're going to put our plasmid into a cell. So when we do that, that is transformation. So we are going to introduce the plasmid to the cell surroundings, and then that is going to get into the cell. So we take our bacteria, we take our plasmid that now contains our insulin gene, if we're saying that we're doing that, and then we're going to introduce that plasmid to the cells. And this is how we most commonly do it. Now, we can't just do this with any cell. We do this with what are called competent cells. So competent cells are those that are ready to take up DNA. And we have different ways of having a competent cell. And these are right here. So we have 3 ways to get competency. First of all, there are naturally competent cells. So some species, like Bacillus species, when you put foreign DNA around them, they just automatically want to grab up that foreign DNA and bring it into their own cell. So this is just naturally competent, and they automatically do this whenever you put DNA around them. We don't often find in laboratories the ability to use naturally competent bacteria. And even if we do, typically it's a pretty low level that they're going to do this at. And so oftentimes they are then going to be made even more competent utilizing either number 2 or 3 here. So number 2 is to make them chemically competent. So we can use chemical treatments. And when we use chemical treatments, we neutralize the charges on the cell membrane, and it weakens that cell membrane. And so then the cell is kind of in this like, oh no stage. And I think we've spoken about it before, when cells are in this kind of frightened stage where they are being adjusted or changed, their neutral or their charges on the plasma membrane are being changed, they then are going to start grabbing up foreign DNA because it's going to be weakened. So their cell is weakened, and we're able to then get DNA into them. Thirdly, we can make them electrocompetent. So electro, meaning electric field here, we are going to expose the bacteria to an electric field. Basically what we do is we kind of shoot holes in their membrane using electricity. So that makes them electrocompetent. So we kind of expose them to this electrical field. It shoots holes or pores in their membrane, and then when those holes or pores are there, then things start to just naturally go into the cell. And when they do, they're going to be repairing their membrane. They're going to be frantically kind of fixing all of these holes. But when they do that, they're going to be bringing in that foreign DNA. So either the foreign DNA will just start to come in on its own, or it'll start grabbing it because it's in a state where it's, in danger. And so it's just going to start grabbing up DNA while it's fixing its plasma membrane. So again, competent cells are those that are ready to take up DNA. We can either have naturally competent cells, or we can have chemically competent cells, and we can just do different types of chemical treatments that we'll talk about, or we can make them electrocompetent, which is that electrical field. So either way, what we're doing, though, is we're applying this plasmid that we have inserted the DNA we want into the plasmid, and then we're forcing these cells to take it up or to pick it up. All right, so we have 2 other ways that we can go through and get the plasmid into the bacteria. So we had our transformation, which is the most common one, which is why we spoke quite a bit about that, and the different ways to make cells competent and get that DNA into the cell. But we can also use transduction and conjugation. So transduction is using phagemids, which are plasmids within the bacteriophage DNA. Or with bacteriophage DNA. So this would be using our bacteriophages, and when we are manipulating those bacteriophages or the plasmids within them, then we can use those to infect or to inject their DNA into bacteria. So this one is not used as much. It's a little bit more difficult to manipulate bacteriophages than it is to just make cells competent and get the DNA in there. But sometimes this is the best method for whatever reason. Lastly, we can utilize conjugation. So we can actually get an F plus cell, one that makes that F pilus, and then we can make sure that that F plus cell has the plasmid with the gene we're looking at or we want. And then we can put it in with a lot of F minus cells, and we can allow that F plus cell and then the F plus cells that come from that F plus cell to infect those F minus cells and pass along the plasmid along with it. Because remember, the F plus cell, when it is going to try to infect an F minus cell, it creates that F pilus, and then it's going to start sending that DNA over into the F minus cell. And oftentimes it's going to be sending not only the plasmid with the F pilus genetic information, but also any other plasmids that it has. So on the right-hand side here, again, this is kind of all of our molecular cloning steps that you need to be familiar with here. So if we take our foreign DNA up here at the top, take our plasmid, we cut both of those with the same restriction enzyme. We can get either sticky ends or blunt ends when we do that. Then in the second step here, we are going to hybridize. So in hybridizing, remember that that's going to allow those nucleotides to anneal and allow those backbones to ligate. So we add our DNA ligase that's going to reattach those DNA backbones. So up here, and they kind of separate it out here. So up here they're talking about annealing because that's going to happen first. Annealing will happen because, again, we have these ends that just want to hydrogen bond. So when we put them in, it hydrogen bonds, but then they're showing here this is where DNA ligase is going to go in and fix up those backbones. Then we can utilize transformation, transduction, or conjugation to get these now recombinant plasmids, right, because this is recombinant DNA now because it has DNA from two different organisms, and get those into the bacterial cell. Now here they're going through the process of sometimes this doesn't work, right? So some bacteria don't take up the plasmid, and so then they don't have the plasmid in here, and therefore are not ampicillin resistant, because remember this plasmid in this example also has ampicillin resistance. The second one is showing this is where the bacterium takes up the non-recombinant plasmid with the intact LAC-Z gene. So we're saying that this one has ampicillin resistance, and it also has the LAC-Z gene in it. But in this case, you can see it doesn't have any red, and that's because for whatever reason, remember that when it's cut up here, these sticky ends really want to attach. So maybe they just attached right back together and then stayed together as its regular plasmid. They didn't get any of this DNA over here. So then we have some bacteria that take up the non-recombinant plasmid, so the LAC-Z gene is intact. It also has the ampicillin resistance gene, or the third option is that the bacterium actually takes up the recombinant plasmid and therefore is not able to make the enzyme to break down XGAL. So you can see here that it actually has that in there, and then it no longer has an intact yellow section, which is, you can see here, the LAC-Z gene along with the restriction enzyme site. So then as we spoke about here, we can do blue-white screening. So remember that the blue colonies have the non-recombinant plasmids because they have an intact LAC-Z gene, so they're able to make the beta-galactosidase enzyme, but we can just say the LAC-Z enzyme. And so since it can make it, it can degrade that XGAL. Since it can degrade the XGAL, it actually turns the cells, that colony, blue. So we know that the blue ones didn't get our, say, insulin plasmid, or insulin gene in the plasmid. Now then the white colonies have the recombinant plasmids, and we know that because we've disrupted that LAC-Z gene. If we've disrupted that LAC-Z gene, it can't make that enzyme anymore, so it can't break down XGAL. It doesn't get hurt by it, but it can't use it as an energy source, so then it's a white colony. It doesn't turn itself blue. So then we know, okay, these cells, these white cells, are the ones that have the plasmid that I want because it has broken up that LAC-Z gene, it has the insulin gene in it, so then you can, take a scraping of this particular colony here, put it in some broth, and grow whole new cultures that all have the insulin gene in it because you know that you got a colony that has the insulin gene in it. All right, so utilizing this information now, we can go about doing this process and getting different things at the end of it. So one of the things that we can do with this molecular cloning process is create a genomic library. So as it says here, a genomic library is a complete copy of an organism's genome, and that's contained as recombinant DNA plasmids that are engineered into unique clones of bacteria. So if we look at the picture down here at the bottom, it says DNA is extracted from the organism and cut into fragments, right? So this is kind of like the example with the insulin gene, except what we're saying is we're going to take this particular organism, whatever this is, and we're going to chop up its DNA. So we cut it up into fragments, utilizing whatever restriction enzyme we want to use. Then we take the plasmid, we cut it up with the same restriction enzyme, and then what we're going to do is we're going to combine those. We're going to hybridize the DNA fragments with the plasmid, excuse me. So the DNA fragments are inserted into the plasmid, but since we cut it with this restriction enzyme, we're not looking for a specific gene. What we're doing is we've chopped up the entire genome, and then we are going to insert all of these sections into the plasmid. So some of the plasmids are going to get whatever this red gene is, some are going to get the yellow, some are going to get the green, and then we're going to take these plasmids and transform them. So the bacteria transformed with the vectors, so putting that DNA, those plasmids with the DNA into the bacteria, and then we can grow those bacteria. They're going to replicate and produce colonies, and then we have lots and lots of bacteria that have this chunk, lots of bacteria that have this chunk, and lots of bacteria that have this chunk. And what this does is it allows for large quantities of each fragment. So in this case, then in a laboratory, if we were making a genomic library for this organism, right? So a library, meaning that this is all of the information that we're going to get on this organism genomically, right? Their genes. What we can do is we can then take this bacteria here and keep it in a test tube, and then we have the ability to utilize whatever information is encoded in this DNA. Same thing with over here, same thing with over here. So we have these chunks of DNA, and then we can use them. So one way that we can use them is to determine the sequence of DNA within those plasmids, and then the function of any of those genes that are present. So we can utilize this again as a sort of library. We can go in and we can say, okay, we're going to use this fragment. First of all, we can go in and we can find out the sequence of the DNA chunk that we put in there because we know what we cut the restriction enzyme with, or what restriction enzyme we use to cut it. So we can figure out what nucleotides are between those cuts. So we can come up with all of the sequence of DNA for this particular organism. But then also the function of those genes. So we can see the function either in the bacterium themselves, or we can utilize that information to somehow determine the function of any of the genes that are present in that chunk. So then here is kind of the steps that we talked about here. Cut the DNA and plasmid with the same restriction enzyme. The bacteria takes up a single plasmid, then we grow colonies of those cells, and then these clones all carry the same recombinant plasmid. So that's what these are right here. All carrying the same recombinant plasmid, and then we can keep them and we can use them later. And that's why it's called the library, because then we'll go through and all of these individual chunks will all be stored in long-term storage, and then they can be utilized separately when they need to. We can also see we can do this with bacteriophages as hosts. So these are going to hold a larger amount of foreign DNA. So we can use here, we have our cellular genome, we have the phage genome. Again, we use the same restriction enzyme. We cut up the cellular genome, cut up the phage genome. Then we build a recombinant phage, or phage, which is similar to what we just spoke about with the bacteria. We build that recombinant phage, and then we can infect E. coli with that recombinant phage, and then we can grow it, and then we see plaques. This plaque contains phages from that unique fragment from the original genome. So again, we're chopping up the whole genome here. We're cutting up into pieces. We're getting, this one has A, this one has B, this one has C, and then when we go in and we place these in with a bacterial lawn, the phages are going to go in and they're going to insert themselves into the bacteria. They're going to go through the lytic cycle, and then that means that what they're eventually going to do is burst the cell, kill the cell, and then in these plaques, which are areas that don't have bacteria, then we have each of these plaques will contain these phages with these different fragments. So we can see that kind of stepwise up here. Bacteriophages as hosts, so they hold larger inserts of foreign DNA. We can do smaller number of cultures to clone all of them. We have our recombinant phage DNA, which is similar to the recombinant DNA we've already spoken about. It's made by ligating digested phage particles with the genome, like we said, and then we package it into phage particles and allow it to infect a bacterial lawn, and then each one of these plaques is going to give us a different molecule, so a different recombinant DNA molecule. And then we can screen that for the gene of interest. So again, it's a way to come up with a genomic library. So then we have, all of these phages have this gene in them, all of these phages have this gene, all of these phages have this other gene. And we can go in and we can utilize that information for whatever purposes. So down the line, if we find out that this organism can do XYZ and we need that for our own humankind, then we can go into a genomic library and we can get that DNA out of there and use it for testing and for research. So similar to the DNA libraries that we just spoke about, we can also make what's called a cDNA library. So the C stands for complementary DNA, so cDNA library. And this is where we're going to use the mRNA molecules, and then we are going to figure out from those mRNA molecules, we are going to determine the expressed genes of an organism. So in this case, what we're doing is we're not taking its specific DNA and chopping it up into pieces and then making a recombinant DNA with it and then putting that DNA in test tubes so that we can use that DNA later and figure out what it's for. Instead, we're kind of doing a backwards situation here. We're using the mRNA, right? And remember, mRNA is transcribed from DNA. So we're utilizing the mRNA molecules to focus on the expressed genes of an organism. So what genes that organism is expressing. And so remember that organisms have all of their DNA in every cell of the organism. But some cells are only going to express some things. So a good example is like humans, right? So every single cell in our body has every single bit of DNA. Every single cell has the same DNA. But not all of our cells express the same genes, right? And so our pancreas, in this example, expresses the genes for insulin. Our eyeballs don't express the genes for insulin. And so on and so forth, right? So the reason that our skin cells look like skin cells is because they express different genes. And so therefore our skin cells have mRNA to make things like keratin, for example, or melanin, or any of the things that we find in a skin cell. However, other cells, like the example here, pancreas cells, are going to go about doing other things, like making glucagon or making insulin. They're not going to be concerned with making keratin, for example, or melanin, which is something skin cells do. So each cell is going to be expressing different genes of the same DNA. So we're working backwards to say, okay, if we take, let's say, a pancreas cell from a human, we get all of its mRNA, we're going to work backwards to then get the DNA that's coming from that mRNA, or a skin cell, or any other thing. So we use the mRNA to work backwards to determine what genes are expressed, because in this case, we can't clone RNA directly. And even if we could clone RNA directly, what we likely want are the genes that are associated with that mRNA anyway, because we want to know the actual genetic information that we can clone and use for purposes of recombinant DNA, for example. So our mRNA is a template then for reverse transcriptase. So this is where we have utilized viruses that have reverse transcriptase, like HIV. We have taken what they do, and now we're using it for good in biotechnology. So we utilize the mRNA template. We put in some reverse transcriptase. So you can see here the RNA is extracted from the organism. We utilize reverse transcriptase. It's going to build cDNA. And again, the C is just saying it's complementary DNA. So it's not direct DNA, but we're making the DNA from that mRNA. So then the rest of the steps are pretty similar, right? We've built the cDNA, so now the cDNA is just used like our chopped up DNA previously. Put into a plasmid, inserted in, so once they're inserted into the plasmid, then we put them into the bacteria, and then we clone the bacteria, and we have our genomic library, which in this case is a cDNA library. And that is, then we can see what these cells are expressing. So the different genes. And we can see this in our prokaryotic organisms with our Serratio marquescens, right? So maybe somebody took the Serratio marquescens that was grown on the desk in the laboratory that has a different phenotype. Remember, that ends up looking pink at about 30 degrees C versus the Serratio marquescens that was growing at 37 degrees C. Once it's growing at 37 degrees C, we don't see that pink. So maybe the Serratio marquescens growing at 30 degrees C, they went through and they did a cDNA library and they found out what gene is it that's turned on at 30 degrees C versus it's not turned on. We don't have mRNA for it. It's not expressed at 37 degrees C. And then we have that information for whatever purpose might be useful. All right, so thus far we have been talking about prokaryotic organisms, but we can do this with eukaryotic organisms as well. And I kind of started the talk about this because what we were really talking about with genetically modified organisms is transgenic plants. So in the case of eukaryotes, we can do transfection. Transfection is when we introduce recombinant DNA into a eukaryotic organism. Instead of transformation, we're transfecting. When we do that, we get something like transgenic plants. So, for example, a delayed-ripening tomato. We do this a lot with plants. This is where we get the GMO thing from, the genetically modified organism, is because we're making transgenic plants oftentimes when we're talking about food sources. Also, this is done with livestock. So we have put DNA into goats, for example, that can then secrete different pharmaceuticals in their milk so that we can put this into the organism. Then they make that, whatever that product is for us, that we can take the milk and then we can separate it out of the milk and we have lots and lots of whatever that particular gene is. However, it's a little bit more complicated. If we're doing studies with genetic information, that's why oftentimes we use things like prokaryotic organisms, like bacteria. Even when we're talking about something like insulin that we're making for humans, it's more difficult to do this with eukaryotic organisms. And that's because, first of all, they're not competent. So it's harder. Our cells don't just automatically try to grab DNA whenever it can. Also, they don't have plasmids, right? So eukaryotic organisms, their cells, don't have extra chromosomal DNA in the form of plasmids just hanging out. So if we try to stick a plasmid into a human, our cells aren't just going to try to grab up DNA, especially because plasmids don't mean anything to our cells. So it's a little bit more difficult. However, we do have techniques to overcome this. One technique is electroporation. This is kind of similar to electrocompetency because in this case we're doing something similar where we're going to put this electrical pulse, and putting this electrical pulse is going to basically poke holes in the DNA, or not in the DNA, I'm sorry, in the plasma membrane. When we poke holes in the plasma membrane, then we can force DNA to get into the cell. Because before, when we said we were going to do electrocompetency, remember we poked holes in the plasma membrane and then the cell automatically is going to suck up DNA and then repair itself. But again, we don't have competent cells in eukaryotic cells. So this pulse is going to not only create pores, it also is going to generate a positive charge on one side of the cell. You can see that here. And on the cell's interior, and then negative on the other side. And so what happens is DNA is negative, right? So DNA, the charge is negative. If we make the inside of the cell positive, then our negatively charged DNA wants to go to the positively charged area, and so then that's going to allow the cell to bring the DNA into it. And then we want to put the cell in an environment that's going to heal itself, and it'll fix those holes. So we apply that pulse. Those pores are going to be there. That's going to allow the DNA to be attracted to the positive side, which means that the DNA gets into the cell. And then after the pulse, then these pores are going to be sealed up, and then the DNA is inside the cell. Another technique that we can use is called microinjection. So this is injecting DNA into cells using a micropipette. You can see an image of that on the right side here. This is our microinjection needle. And we see this a lot with in vitro fertilization as an example. So again, with in vitro fertilization, we're taking one cell and we're putting the DNA of another cell into it. So this is a type of genetically modifying, genetically modifying something. It's not recombinant DNA, but we're taking DNA from one organism, taking DNA from another one and putting them together. So microinjection is one way of doing that, which is basically we just take a pipette or a needle and we inject it, as the name implies. Poke a hole in the plasma membrane, put the DNA inside, and then take that out, and then the plasma membrane will repair itself. Another method are gene guns. So in this case, we're shooting gold or tungsten particles. But those gold or tungsten particles are coated with DNA. And so this is something that's often used in making plants genetically modified. So you can see at the bottom here, we have our gene gun. This is what it actually looks like, but this is inside of it down here. And so we can take our gold or tungsten particles, and previously we're going to incorporate DNA with that. So then the DNA, it'll likely be charged up gold, and then we have our charged DNA, so they're going to attach to each other. And then we can fill this gene gun up, and then basically we just shoot it at the cells. When that happens, just like guns do, they'll go right in through that plasma membrane, and then that will get into the cell. So first of all, these plant cells are actually protoplasts. So we're not just taking like a green leafy plant here and then shooting it with a gun. What we need to do first is the cells have to be treated. So we actually take plant matter. They're treated with enzymes that dissolve that cell wall that's going to protect the cells. After that cell wall is gone, when it's just the plasma membrane, that's when it's called a protoplast. Now that we have no more cell wall, we're able to shoot these little gold or tungsten particles into the plant cell because it's going to go through that cell membrane. We don't have a cell wall anymore. And then that's going to get DNA into the protoplast. And then, of course, it's going to heal itself. So it'll start to build that cell wall again. It'll fix its plasma membrane. And now we've gotten that DNA into a plant cell. We can also use something called shuttle vectors or viral vectors. So first, our shuttle vectors are plasmids that move between bacterial and eukaryotic cells. This is very, very specific, though. So oftentimes, this is when we're using a TI plasmid. Here, a TI plasmid is a tumor-inducing plasmid. So we see this happen a lot of time. We see this in Agrobacterium genus. So we can take the tumor-inducing plasmid, the TI plasmid, and put it in Agrobacterium tumefaciens. That Agrobacterium tumefaciens specifically is a bacteria that causes little tumors. It causes tumors on the roots of a plant. So in the roots of a plant, it'll attach to the plant, and then it'll start to grow these little tumors on the plant. Typically, these tumors don't hurt the plant at all. It's a symbiotic relationship where the bacteria are going to be making things that the plant needs. Think back to our nitrogen cycle that we were talking about. And then the bacteria has a place to live and gets nutrients as well. So our Agrobacterium tumefaciens that makes these little root nodules, we can utilize those bacteria then, because when we put this plasmid into it with our recombinant DNA, with whatever it is that we want to get into our plant cell, those organisms, now the plant and the bacteria, start to share DNA because they're so closely linked. And when they do this, then we can get that DNA into the plant. So we can remove, as it says here, remove those tumor genes, and then we can insert our gene that we're looking for. So we take our Agrobacterium tumefaciens, we can isolate the plasmid, we can take this region here and insert our gene of interest, whatever that is. So we clone the gene of interest, put it into the TI plasmid. Now it's a recombinant plasmid. Then we put it back into Agrobacterium tumefaciens, and then we can infect the plant with the Agrobacterium tumefaciens. And then now that bacteria is going to share DNA with the plant. And therefore, we hope that the DNA will then move into the plant. And once it does, then that will be inside of the plant cells and be made copied over and over again. Another one that we already mentioned slightly was a viral vector. So a viral vector is where we can go in and we take out the viral genes, so the things that make it virulent. We can use a virus because we know how a virus works, right? It's going to get into the cell. So it always goes into the cell. It's an obligate intracellular organism. And since it's going to get into the cell and typically cause an infection or something, disease in an organism, instead we take out all the genes that have the disease part, and instead we insert our genes of interest. So it still acts like a virus in the fact that it has to go inside of an organism in order to live, and it'll hijack its machinery. But in the case of it hijacking its machinery, it's no longer going to be hijacking it to make its virulence, proteins or toxins, whatever it is. Instead, it's going to be making copies of whatever the gene is that we put in there. So then we can infect the host cell there, and then we have lots of copies of that being made, or depending on the type of virus it is, it will then incorporate itself into the DNA of the host. This is actually what we see in gene therapy, is we can use viruses with recombinant DNA. So the virus part per se is taken out, these viral genes, meaning what causes infection, and we can put in things that we want in our organism, in our human, for example. Then when that virus gets into the human cell, it can incorporate its gene, its DNA, into the human cell's DNA, kind of like HIV does. And then that human is now going to replicate this cell that has all of this stuff in it. And that can be used for gene therapy, which we'll talk about in more detail later.